Have you ever paused to consider the silent architects within your body, those microscopic powerhouses we call cells? They perform astonishing feats, from fueling every thought you have to standing as the first line of defense against invaders. And they do all this while maintaining their intricate internal world perfectly distinct from the, well, chaotic environment outside. This isn't just biological magic, it's the profound work of what we might call the unsung heroes of biology, biological membranes. Today, we're embarking on a true deep dive into the incredible world of cellular membranes. Our mission is clear. We're not just scratching the surface here. We're going to rigorously unpack their complex structure, explore their truly mind-boggling array of functions, and uncover why they are absolutely fundamental, indeed indispensable to life as we know it. Our goal for this deep dive is for you to truly master this information, understanding it with depth and clarity expected of elite professionals in the field.
That's precisely our aim. And when you think of membranes, try to move beyond the simplistic idea of just, a simple barrier. Instead, envision them as dynamic, highly fluid, and astonishingly active structures. They are constantly organizing, communicating, and orchestrating nearly every essential process that happens both within and between cells. We will meticulously explore the how and why behind these critical cellular components, drawing deep insights from foundational biochemical and, well, biophysical principles.
And to achieve that level of mastery, that real, profound understanding, we've meticulously curated a fascinating stack of detailed biochemical texts and cutting-edge research excerpts. We've distilled the most important nuggets, the core truths, to give you a truly well-informed, accelerated pathway to this knowledge. So let's begin this journey into the cellular frontier. Hashtag I, the fundamental role of membranes in life. Okay, so let's unpack this foundational concept right from the start. At its most basic, fundamental level, membranes are what quite literally make a cell a cell. The plasma membrane, the outer boundary, it forms a closed compartment. It's not just a wall. It actively defines the cell's physical boundaries, acting like the ultimate, highly intelligent security system, meticulously deciding what gains entry and what must be kept out.
And you're absolutely right. It's profoundly more than just a passive wall. The plasma membrane possesses this exquisite selective permeability. What this means is that it rigorously controls the passage of substances, meticulously maintaining precise and, frankly, drastically different compositions between the intracellular environment, the cell's inside, and the extracellular environment, the world outside the cell. This barrier function isn't merely important, it is absolutely critical for the very existence and survival of life. But its role extends far beyond simply keeping things out. The plasma membrane is intensely active in facilitating the exchange of materials with the extracellular environment. This occurs through remarkable and complex processes like exocytosis, where the cell actively expels substances, packaging them into vesicles that fuse with the membrane.
Right, sending things out.
Exactly. And endocytosis, where it takes things in by engulfing external material into new vesicles formed from the membrane. Moreover, the plasma membrane enables adjacent cells to communicate directly, forming specialized structures known as gap junctions that act as direct cytoplasmic bridges, like little tunnels between cells.
Wow, okay.
And we must also emphasize its crucial roles in broader cell-cell interactions, where cells recognize and adhere to one another, and in the intricate dance of transmembrane signaling, where external signals are received and relayed into the cell's interior, orchestrating its responses. It's constantly busy.
It's clear then that it's not just the external boundary, not just the cell's skin, so to speak. Membranes are equally busy inside the cell, creating these incredible, highly specialized compartments. It's almost like a cellular apartment building, isn't it? With each room, each distinct compartment having a very specific, dedicated purpose.
That's a perfect analogy, actually. Intracellular membranes play an absolutely pivotal role in shaping most of the morphologically distinguishable structures within the cell that we call organelles. Consider the mitochondria, often dubbed the powerhouses of the cell, right? Where most of our ATP is generated.
Yeah, the energy factories.
Or the endoplasmic reticulum, the ER, that vast network vital for synthesis and folding of proteins and lipids. Then there's the Golgi apparatus, which functions kind of like the cell's post office for modifying, sorting, and packaging molecules. You also have secretory granules storing and releasing substances, lysosomes acting as the cell's recycling centers, breaking down waste.
Getting rid of the trash.
Exactly. And, of course, the nucleus, which houses and protects our precious genetic material, the DNA. Each of these membrane-bound structures serves to precisely localize specific enzymes, ensuring that biochemical reactions happen in their designated places without interference. They also function as integral elements in excitation response coupling, for example in muscle contraction, where ion movements across internal membranes trigger the muscle shortening.
Ah, okay.
And critically, these membranes provide the essential sites for energy transduction, whether it's photosynthesis in plant chloroplasts, converting light into chemical energy, or the highly efficient process of oxidative phosphorylation in our own mitochondria, where ATP is generated through an electron transport chain embedded right in that inner mitochondrial membrane.
So why is all this compartmentalization, both inside and out, so fundamentally vital? It ultimately comes down to maintaining a remarkably delicate balance, doesn't it? Especially with water and various ions. It's about creating and meticulously holding on to very distinct internal and external chemical environments.
You've hit on a core principle of life. Life, as we understand it, originated and evolved in an aqueous environment, and consequently, our cellular processes are just exquisitely tuned to function optimally within this water-rich milieu. Roughly 60% of our lean body mass is composed of water, and this water is strategically distributed into two major, functionally distinct compartments. First, we have the intracellular fluid, or ICF. That's the fluid found inside the cells. This compartment constitutes a significant 2/3 of the total body water, making it the largest fluid compartment by far. The ICF provides a highly sterilized, tightly controlled internal environment where a vast array of critical cellular functions are performed. This includes, the intricate processes of making, storing, and utilizing cellular energy in the form of ATP.
The energy currency.
Precisely. Enabling cells to repair themselves from daily wear and tear, facilitating cellular replication, which is essential for growth and tissue maintenance, and supporting all the unique cell-specific functions that define different cell types, from nerve impulse transmission to neurons to hormone secretion in endocrine cells. This is truly the workshop of the cell. Then we have the extracellular fluid, or ECF, which makes up about 1/3 of the total body water. This ECF is further distributed between two subcompartments: the plasma, which is the fluid component of blood circulating throughout our vascular system,
Right, in the bloodstream.
and the interstitial fluid, which directly bathes the cells, filling the spaces between them. Functionally, the extracellular fluid acts as a dynamic two-way delivery and removal system. It constantly brings essential nutrients like glucose, fatty acids, amino acids, along with vital oxygen, various ions, crucial trace minerals, and a wide array of regulatory molecules like hormones.
The messengers.
Exactly, which coordinate cellular functions across the entire body. Simultaneously, the ECF efficiently removes metabolic waste products, including carbon dioxide and other toxic or detoxified materials, from the immediate cellular environment. This ensures a continuously clean and optimal surrounding for cellular activity, truly making it the cell's lifeline and, well, its waste disposal system too.
What's truly fascinating here, and really speaks volumes about the membrane's role, is how wildly different the ionic compositions are between these two fluid compartments, the inside and outside of the cell, and how membranes painstakingly, constantly work to maintain those differences. It's like having two completely distinct chemical soups right next to each other, yet they never just mix haphazardly.
It's a dramatic, precisely maintained difference. If you look at typical mammalian cell concentrations, the internal environment, the ICF, is remarkably rich in potassium ions, K+, and magnesium ions, Mg2+. Its major inorganic anion is phosphate, PO43, and it contains a high concentration of proteins that act as crucial intracellular buffers, helping to maintain a stable pH despite metabolic activity.
Okay, so high potassium, high magnesium inside.
Right. In stark contrast, the extracellular fluid is characterized by a high content of sodium ions, Na+, and calcium ions, Ca2+, with chloride, Cl, being its predominant anion.
And sodium and calcium outside, big difference.
Huge difference. And these enormous ionic differences are not accidental or static. They're meticulously maintained and actively regulated by specialized membrane proteins like pumps and channels, and by the unique lipid compositions found within the various cellular membranes. This precise control over ionic gradients isn't just a detail. It is absolutely fundamental to almost every cellular process, including nerve impulse transmission, muscle contraction, maintaining cell volume, you name it. Without these gradients, life as we know it simply couldn't exist.
So if membranes are so crucial for establishing and maintaining these fundamental balances both within and around the cell, what happens when they go wrong? What are the profound consequences when this intricate, finely tuned system breaks down?
When membrane components are altered, the consequences are indeed profound and far-reaching. Changes in their structure or composition directly impact vital processes like water balance and ion flux across the cell,
Yeah.
thereby disrupting countless essential biochemical pathways and cellular functions. More specifically, localized deficiencies or structural alterations in certain membrane components, most often caused by genetic mutations in the genes encoding specific membrane proteins, can lead to a wide array of severe diseases. We see clear and well-documented examples of this in conditions like familial hypercholesterolemia. That's a genetic disorder of cholesterol metabolism. It results from faulty, low-density lipoprotein, LDL receptors in the plasma membrane, leading to dangerously high cholesterol levels.
Right, the LDL receptors aren't working properly.
Exactly. Another stark example is cystic fibrosis, where a defect in a chloride ion channel disrupts fluid balance in various organs. We'll probably talk more about that one. And then there's hereditary spherocytosis, a blood disorder where mutations in structural proteins of the red blood cell membrane lead to fragile, spherical red blood cells that are easily destroyed. Each of these conditions, among many others, unequivocally demonstrates how critical membrane integrity and proper function are to overall health, and how a seemingly small defect at the molecular level can have devastating systemic consequences.
So, to distill it, basically, if your membranes aren't working right, your cells aren't working right. Normal cellular function in every tissue and every organ critically depends on normal, healthy membranes. It's like the foundation of a skyscraper. If it's cracked or compromised, the entire structure is inevitably compromised. When we delve into the structure of biological membranes, there's one groundbreaking model that stands as the bedrock of our understanding. It paints a remarkably vivid and accurate picture of this incredibly tiny and complex cellular component.
That foundational model is the fluid mosaic model, proposed back in 1972 by Singer and Nicholson. It remains absolutely central to our understanding of membrane architecture and function because it beautifully captures both the dynamic and heterogeneous nature of membranes. The model describes the membrane as integral membrane proteins. These are the workhorses, the functional machinery like icebergs, floating and moving within a fluid sea of predominantly phospholipid molecules.
Icebergs in a sea of lipids. I like that.
It vividly emphasizes both the embedded nature of proteins within the lipid environment and the dynamic mobile character of the lipid bilayer itself. It's not a static, rigid structure, but a constantly shifting, interacting landscape.
And the early evidence for this radical idea was quite clever, wasn't it? Using fluorescently labeled proteins, researchers could actually watch them quickly redistribute in hybrid cells, proving that dynamic fluid nature. They literally watched these molecular icebergs drift.
It was indeed a groundbreaking and elegant experiment that really solidified the model's validity. Researchers artificially fused a mouse cell with a human cell, creating a single hybrid cell. Then they used antibodies tagged with different fluorescent dyes, one color for mouse proteins, another for human proteins, to label specific integral membrane proteins on the surface.
Okay.
Initially, the mouse proteins were confined to one half of the fused cell and the human proteins to the other. But what they observed was truly compelling. Within minutes, these labeled proteins, originally segregated, rapidly and randomly redistributed across the entire plasma membrane of the hybrid cell, mixing completely.
Wow, that fast.
That fast. This rapid mixing was direct, compelling visual proof of the membrane's inherent fluidity and the lateral mobility of its protein components. And to further underscore the fluid part, we know that phospholipids themselves exhibit even more rapid lateral diffusion, moving several micrometers per second within the plane of the membrane. This constant, swift, chaotic movement of lipids is truly a defining feature of biological membranes, enabling their diverse functions.
So let's really dive into the C itself, the lipid bilayer. It's the foundational structure of all biological membranes, and its spontaneous formation in an aqueous environment is quite remarkable. It just organizes itself, almost magically.
It's less magic and more an elegant outcome of fundamental biophysical principles, really. Membranes are sheet-like enclosed structures, typically around 5 to 8 nanometers thick, incredibly thin. Tiny, consisting of an asymmetric lipid bilayer. And they form spontaneously, self-assembling in aqueous environments because their major lipid components, phospholipids, glycosphingolipids, and cholesterol, are amphipathic. This is a critical characteristic. It means they possess both hydrophobic or water-hating and hydrophilic or water-loving regions within the same molecule, like a two-phase molecule in a way.
So one end likes water, the other hates it.
Exactly. And in water, these amphipathic molecules automatically organize themselves into configurations where their hydrophobic regions are shielded from water, while their hydrophilic polar head groups are immersed in the aqueous environment. This spontaneous organization minimizes unfavorable interactions with water and maximizes favorable ones, leading to the formation of structures like spherical micelles.
Like tiny bubbles.
Sort of, yes. Smaller and encapsulating hydrophobic interiors. Or, more crucially for biological membranes, the flat extended bimolecular lipid bilayer. This bilayer is the fundamental structural framework. The primary driving force behind this self-assembly is known as the hydrophobic effect. This effect describes the tendency of nonpolar molecules to self-associate in water, not really because they are attracted to each other, but because their presence forces water molecules into an ordered, cage-like structure around them. By associating, these nonpolar molecules reduce the total surface area exposed to water, which allows the previously immobilized water molecules to be released and become more disordered, thereby increasing the overall entropy or disorder of the surrounding solvent. This increase in the total systems disorder provides the thermodynamic favorability for bilayer formation. It's essentially water pushing the nonpolar parts together, which is quite profound when you think about it.
Now that we understand why they organize, tell us more about these major lipids that actually make up this incredible bilayer. What are they? What are their distinct structures, and what general roles do they play?
Certainly. There are three major classes of lipids that predominantly make up mammalian membranes, each contributing uniquely to the properties of the bilayer. First, and most abundant, are the phospholipids. The most common type are phosphoglycerides, which are derived from glycerol. They feature a glyceryl phosphate backbone to which two fatty acids are attached by ester linkages, and a phosphate group that is further esterified to a polar alcohol. A key intermediate in their formation is phosphatidic acid, but in most membranes, this phosphate is linked to an alcohol like choline, ethanolamine, glycerol, inositol, or serine. For instance, phosphatidylcholine is usually the major phosphoglyceride by mass in human cell membranes. It's a key component due to its sort of cylindrical shape and deuterionic nature.
Meaning it has both positive and negative charges.
Right, charges that balance out. The second class of phospholipids is sphingomyelin. Unlike phosphoglycerides, sphingomyelin has a sphingosine backbone, which is a long-chain amino alcohol rather than glycerol. A fatty acid is attached by an amide linkage to sphingosine, forming a molecule called ceramide. When the primary hydroxyl group of sphingosine is then esterified to phosphorylcholine, sphingomyelin is formed. As its name suggests, it is particularly prominent in myelin sheaths, which insulate nerve fibers, facilitating rapid electrical transmission. The second major lipid class is the glycosphingolipids, or GSLs. These are distinguished by being sugar-containing lipids, also built upon a ceramide backbone. Examples include cerebrosides, which contain a single sugar unit, often glucose or galactose attached to the ceramide, and gangliosides, which contain more complex branched oligosaccharide chains, sometimes up to 7 sugar residues attached to the ceramide.
So they have sugar chains sticking out.
Exactly. These GSLs are primarily found in the plasma membranes of cells, with their sugar components consistently displayed to the exterior of the cell, forming part of the glycocalyx, or sugar coat. This external positioning is incredibly crucial for various cell recognition processes, cell adhesion, and acting like cellular ID tags or antennae, allowing cells to interact with their environment and with each other. They are involved in everything from immune responses to brain development. Finally, we have the sterols. The most common sterol in animal cell membranes is cholesterol. The vast majority of cholesterol resides within plasma membranes, contributing to their structural integrity and fluidity. But smaller amounts are also found in mitochondrial, Golgi complex, and nuclear membranes. Cholesterol has a unique, rigid 4-ring structure. It intercalates or wedges itself among the phospholipids within the bilayer, with its small hydrophilic hydroxyl group positioning itself at the aqueous interface, while the rest of its bulky hydrophobic molecule is buried deep within the nonpolar core of the lipid bilayer leaflet.
Sort of like a little rigid spacer.
Kind of, yes. And it's an important point for comparative biology. Cholesterol is not present in plants. They utilize other plant sterols like phytosterols, which fulfill similar roles but have distinct structures.
And the physical structure of those fatty acid tails, whether they're straight or bent, has a huge impact on how fluid the membrane is, right? It seems like a subtle detail with major consequences for cell function.
Absolutely. It's a critical determinant of membrane fluidity, which in turn profoundly influences membrane function. Saturated fatty acids, lacking double bonds in their hydrocarbon chains, form relatively straight, extended tails. This allows them to pack very tightly together, maximizing van der Waals interactions between adjacent chains. This tight packing leads to a more rigid, sort of gel-like membrane structure at lower temperatures.
Packed tightly, less fluid, makes sense.
Exactly. In stark contrast, unsaturated fatty acids, especially those prevalent in biological membranes, typically exist in the cis configuration, which introduces a distinct kink or bend in their hydrocarbon tails.
A kink, like a bent elbow.
Precisely. The more double bonds an unsaturated fatty acid has, the more kinks it will introduce. These kinks physically prevent tight packing of the lipid tails, which in turn leads to greater spacing between the polar head groups and more room for movement, creating a looser, more disordered arrangement. Consequently, membranes with a higher proportion of unsaturated fatty acids exhibit increased fluidity at a given temperature. Now, cholesterol plays a fascinating and crucial role as a fluidity buffer in mammalian membranes. Its rigid planar structure allows it to interact with the fatty acid tails in a nuanced way that depends on the temperature. Below the membrane's transition temperature, TM, that's the temperature where the membrane shifts from gel-like to fluid cholesterol, interferes with the tight packing of the hydrocarbon tails. By inserting itself between the tails, it effectively disrupts their ordered crystalline state, thus increasing membrane fluidity.
So it makes it more fluid when it's cold.
Right. But at temperatures above the tenium, when the membrane is already quite fluid and disordered, cholesterol's inherent rigidity limits the excessive movement and disorder of the fatty acid tails. It essentially prevents the membrane from becoming too liquid or leaky, thus decreasing fluidity and maintaining optimal viscosity.
So it stops it getting too sloppy when it's warm, a buffer, like you said.
Exactly. It buffers its fluidity. This ability to maintain an optimal fluid state across a range of temperatures is vital for proper membrane function, influencing everything from its permeability to the lateral mobility and function of integral proteins. We even observe this sophisticated adaptation in nature, with bacteria adjusting their lipid composition, or unsaturated fats in the cold, for example, to maintain appropriate membrane fluidity in response to temperature changes in their environment. It's a beautiful example of biochemical acclimation.
Okay, so the lipid bilayer is designed to be a selective barrier. What types of molecules can actually slip through it relatively easily, and what molecules really struggle or are essentially blocked without help?
You're right, the lipid bilayer is remarkably selective. It acts as a highly effective barrier that largely prevents the unregulated passage of most water-soluble and charged molecules. However, there are specific categories of molecules that can readily traverse it. Small uncharged molecules like gases oxygen, O2, carbon dioxide, CO2, nitrogen, N2, along with small lipid-soluble molecules like steroid hormones, estrogen, testosterone, for example.
Things that like fat.
Exactly. And many therapeutic drugs, which are often designed specifically to be lipid-soluble, can readily diffuse directly through the hydrophobic core of the membrane. This is because they have minimal or no charge and are soluble in that nonpolar interior, allowing them to effectively dissolve through the lipid environment. Water itself, quite surprisingly, has a relatively high permeability coefficient across the lipid bilayer. While polar, its small size allows it to sort of sneak through, even though it's not strictly lipid-soluble. The real challenge arises for charged molecules, especially ions like sodium, No+, potassium, K+, calcium, Ca2+, and chloride, Cl ions, as well as larger uncharged polar molecules like glucose.
Sugar, yeah.
Their movement across the bilayer by simple diffusion is incredibly slow, by many orders of magnitude, making it functionally negligible. The primary reason for this is that in an aqueous environment, these ions are hydrated. They acquire a shell of water molecules tightly associated with them due to strong electrostatic interactions.
Like a little water coat.
Exactly. This hydration shell effectively makes the ions much larger and, crucially, significantly reduces their solubility in the hydrophobic core of the lipid bilayer. To strip off this hydration shell and then reform it on the other side would require a prohibitive amount of energy. This is precisely why specialized protein structures, the transporters and ion channels we'll discuss in detail, are absolutely essential for their efficient, rapid, and controlled movement across biological membranes. They need help.
And what about the inside versus outside of the membrane itself? Is it a symmetrical structure, or are there fundamental differences in composition between the two sides?
No, not at all symmetrical. Membranes are profoundly asymmetric structures, meaning their inner and outer surfaces, or leaflets, are chemically and functionally distinct. The asymmetry is not accidental. It is established during membrane biogenesis and meticulously maintained because it is crucial for proper function. For example, integral membrane proteins have a unique and specific orientation within the bilayer. Their asymmetric distribution is precisely determined during their biosynthesis in the ER and subsequent transport. Furthermore, the carbohydrates that are covalently attached to membrane proteins forming glycoproteins and glycolipids are exclusively located on the external surface of the plasma membrane.
Only on the outside.
Only on the outside. This forms a unique sugar coat, or glycocalyx, on the cell surface that is crucial for cell recognition, adhesion, and interaction with the extracellular matrix. There's also a clear asymmetry in phospholipid distribution across the bilayer leaflets. Choline-containing phospholipids like phosphatidylcholine and sphingomyelin are predominantly found in the outer leaflet, facing the extracellular environment. In contrast, aminophospholipids like phosphatidylserine and phosphatidylethanolamine are preferentially located in the inner leaflet, facing the cytoplasm.
So different lipids prefer different sides.
Yes, and this asymmetry is maintained because the flip-flop, or transverse movement, of lipids from one leaflet to the other is an extraordinarily slow process. Half-lives on the order of days or even weeks without help. For proteins, transverse movement is practically non-existent.
They don't flip across.
Almost never. However, specialized enzymes, known as translocases or flippases and floppases, actively transfer certain phospholipids across the bilayer, helping to establish and maintain this precise distribution. Additionally, specific proteins that preferentially bind individual phospholipids, along with phospholipid exchange proteins, contribute to this intricate asymmetry, ensuring the membrane-sidedness is perfectly preserved for its diverse roles.
If lipids form the sea that provides the framework, then proteins are undoubtedly the icebergs within that sea, and they truly do the vast majority of the heavy lifting when it comes to membrane function. They are the active players, aren't they?
That's an excellent and accurate analogy. Membrane proteins are indeed the major functional molecules of membranes, performing an incredibly diverse and vital range of roles. They act as enzymes, driving biochemical reactions localized to the membrane, as pumps and transporters, moving specific molecules across, as channels, forming selective pores for ions, as structural components, providing integrity and shape, as antigens for cell identification, and as receptors receiving signals. Because every type of membrane, plasma membrane, mitochondrial membrane, nuclear envelope, possesses a different and specialized complement of proteins, there's no single typical membrane structure. The lipid bilayer essentially acts as the solvent for these proteins, providing the necessary environment. And we classify these crucial proteins into two main types based on how they associate with the bilayer: integral and peripheral. Let's first examine integral proteins. These are proteins that are firmly embedded within the lipid bilayer, interacting extensively with the hydrophobic tails of the phospholipids. Consequently, they require strong detergents, like SDS, to be released from the membrane for study.
Need something harsh to get them out.
Exactly. Many integral proteins are transmembrane proteins, meaning they span the entire bilayer, often multiple times, weaving back and forth like a thread through fabric, creating transmembrane helices, or sometimes beta barrels. These proteins are usually globular and amphipathic, possessing hydrophobic regions that traverse the bilayer and hydrophilic regions that protrude into the aqueous environments on either side.
So parts like the lipids, parts like the water?
Precisely. Think of transporters or ion channels weaving across the membrane, or simpler proteins like glycophorin A in red blood cells that cross only once. Their asymmetric orientation, which part faces inward or outward, is precisely established during their biosynthesis in the ER, ensuring their functional domains are positioned correctly. In contrast, peripheral proteins do not interact directly with the hydrophobic core. Instead, they bind loosely and transiently to the hydrophilic regions of integral proteins or to the polar head groups of the phospholipids on either the inner or outer surface.
So they're just kind of sitting on the surface?
Essentially, yes, or attached to an integral protein. Because they're not deeply embedded, they can be released relatively easily, for example by treatment with high salt concentrations, which disrupts the electrostatic interactions holding them there. A classic example is ankyrin, which binds to the inner side of the integral band 3 protein in the red blood cell membrane. Ankyrin, in turn, links to spectrin, a key cytoskeletal protein that forms a network just beneath the plasma membrane, providing mechanical support and helping maintain the red blood cell's characteristic biconcave shape.
Ah, so they can link the membrane to the cell's internal skeleton.
Exactly. It shows how peripheral proteins can serve as crucial links, influencing cell shape and rigidity.
And like everything in the cell, these components aren't static, are they? It's truly not a fixed, rigid structure. The whole idea of fluid mosaic suggests constant motion.
Indeed, static is the absolute antithesis of biological membranes. Membranes and their components are remarkably dynamic. Both membrane lipids and proteins undergo continuous turnover, meaning they are constantly being synthesized and degraded, although the rates vary depending on the specific molecule and the cell's needs. In some cases, entire sections of membrane can turn over even more rapidly than individual components, like during active endocytosis and exocytosis. Furthermore, as we discussed with the fusion experiments, lipids and many integral proteins exhibit rapid lateral diffusion, moving freely and quickly within the plane of their respective membranes. This lateral mobility is absolutely crucial, allowing components to interact, rearrange, and form temporary complexes as needed for signaling or transport. However, it's critically important to distinguish this from transverse movement, or flip-flop, moving from one leaflet to the other.
The flipping across we talked about.
Right. For lipids, this flip-flop is extraordinarily slow, essentially negligible for rapid changes. For membrane proteins, transverse movement is practically non-existent. This maintains that crucial asymmetry. A fascinating aspect of this dynamism is protein lipidation. This is where some proteins are anchored to one leaflet of the bilayer by strong covalent linkages to specific lipids.
So the protein gets a lipid tail attached.
Exactly, or other lipid modifications. This can happen at the protein's ends or even internally. Common examples include isoprenoylation, cholesterolylation, and the attachment of glycophosphatidylate linosylate GPI anchors. These lipidation events are highly specific and play absolutely crucial roles in the precise localization, function, and signaling capabilities of these proteins, often acting as dynamic molecular switches.
Knowing all this intricate detail about natural membranes, it's a testament to scientific ingenuity that researchers have actually been able to create artificial membranes. And these artificial membranes have become remarkably powerful tools, haven't they? Not just for research, but also for potential therapies.
They absolutely have, and their development truly showcases our understanding of membrane biophysics. Artificial membrane systems, commonly referred to as liposomes, are spherical vesicles meticulously prepared from mixtures of phospholipids. These phospholipids, being amphipathic, spontaneously form a bilayer that encloses an aqueous interior, perfectly mimicking the fundamental structure of cellular membranes. They are typically prepared using techniques like sonication, using sound waves, or extrusion through porous filters, which break down larger lipid aggregates into these smaller, uniform vesicles.
So, what are the distinct advantages of using these artificial membranes? How do they help us understand biology better or even develop new treatments?
The utility of liposomes is quite significant and multifaceted. Firstly, they offer controlled composition. Researchers can precisely vary the lipid content, the ratio of saturated to unsaturated fats, the amount of cholesterol, specific head groups, you name it.
So you can build the membrane exactly how you want it.
Precisely. This allows systematic study of how different lipid compositions impact specific membrane functions, like fluidity, permeability, or protein activity. It's invaluable for dissecting mechanisms. Secondly, they facilitate protein reconstitution. Purified membrane proteins, often tricky to study once pulled out of their natural environment, can be incorporated into these artificial liposomal vesicles. This lets scientists assess their functional requirements. Does a transporter need specific lipids? Can a receptor signal on its own? Thirdly, the environmental control is unparalleled. You can rigidly control both the internal and external environments, ion concentrations, pH, ligands, in a way that's impossible with living cells. This provides a powerful system for studying isolated membrane processes. And perhaps most excitingly, liposomes have immense potential for drug and gene delivery. When liposomes form, they can entrap various compounds inside drugs, chemotherapy agents, even genes like DNA or RNA.
Like tiny delivery trucks.
Exactly. There's a significant ongoing interest in developing targeted liposomes by adding specific components to their surface, like antibodies, that recognize specific cells, like tumor cells.
So you can send the drug right where it needs to go.
That's the goal. This could potentially revolutionize drug delivery by minimizing side effects and increasing effectiveness. Furthermore, DNA or RNA inside liposomes seems better protected from degradation, making this a promising avenue for gene therapy. It's a very active area of research.
Now, this is truly where the rubber meets the road, isn't it? How cells meticulously manage what comes in and what goes out. It's definitely not just a free-for-all, a chaotic flow. There are incredibly specific, regulated rules governing every molecule's passage across the membrane.
You've captured it perfectly. The movement of molecules across cellular membranes is highly regulated and absolutely fundamental. Generally, we differentiate between two broad categories: passive transport and active transport. Molecules can move passively down an electrochemical gradient by simple diffusion or by facilitated diffusion. Neither of these requires direct energy input from the cell. It's like rolling downhill. In stark contrast, active transport always moves solutes against an electrochemical gradient, from lower to higher concentration, or against an electrical potential. This uphill movement inherently demands a direct expenditure of cellular energy, usually from ATP hydrolysis. Let's elaborate on simple diffusion first. This is the most basic form, the spontaneous passive flow of a solute from higher to lower concentration, driven purely by random thermal movement. The net rate depends on a few things: the molecule's thermal agitation, the steepness of the concentration gradient, and crucially, its solubility in the hydrophobic core, its permeability coefficient.
How easily it dissolves in the lipid part.
Exactly. Gases like oxygen and CO2, being highly lipid-soluble, diffuse readily. Lipid-soluble hormones and many drugs also cross easily. Water, surprisingly, sneaks through reasonably well too, due to its small size. Now, for electrolyte ions like Na+, K+, ClAc, their movement by simple diffusion is dramatically slower. Remember that hydration shell?
The water coat, yeah.
It makes them effectively larger and much less soluble in the hydrophobic core. So simple diffusion for ions is functionally negligible. Other factors affecting net diffusion include the electrical potential across the membrane. Cell interior is usually negative, attracting positive ions, hydrostatic pressure, and temperature. Then there's facilitated diffusion. This is also passive transport down a concentration gradient, but it's mediated by a specific protein transporter embedded within the membrane.
So it still doesn't need energy, but it needs a helper protein.
Correct. The protein involvement introduces specificity and saturation, but no direct energy cost. Finally, active transport is that vectorial movement against a gradient, requires energy, usually ATP, and always involves a specific transporter protein, often called a pump. This is how cells maintain those dramatic ionic imbalances we talked about earlier.
So when we talk about these protein-mediated movements, what's the big functional difference between a transporter and an ion channel? They both move molecules, but they seem to have very different approaches, right?
This is a really key distinction, defining how different molecules are moved and, importantly, the speed. Transporters, also called carriers or permeases, operate by binding the specific solute they move. Upon binding, the transporter undergoes a conformational change, a shape shift, which physically moves the bound solute across the membrane, releasing it on the other side.
Like a revolving door, one at a time.
Exactly like a revolving door. This process is relatively slow because it requires these structural shifts. Transporters are involved in both facilitated diffusion, passive, and active transport. Their rate is typically much slower, maybe 100 to 1,000 molecules per second per protein. Ion channels, on the other hand, function quite differently. They form selective, pore-like structures directly through the membrane, creating a continuous hydrophilic path.
More like an open tunnel.
A selective open tunnel, yes. These pores allow ions to pass through at significantly faster rates, approaching the limits of free diffusion, up to 10 million or even 100 million ions per second per channel.
Wow, that's way faster.
Orders of magnitude faster. Because they simply open a pathway, like a gate, ion channels are involved only in passive transport, always allowing ions to move down their electrochemical gradient. So transporters are specific revolving doors, channels are super fast, selective tunnels.
Let's talk about the energy-demanding active transport systems. Maintaining those incredibly steep electrochemical gradients must be a massive energy drain on the cell, consuming a lot of its resources.
It absolutely is, and it underscores just how vital these systems are. Maintaining those gradients is so critical for everything from nerve impulses to nutrient uptake that it consumes roughly 30% to 40% of a cell's total energy expenditure. That's a staggering amount of ATP.
Almost half the energy budget just for pumping things?
Pretty much. The energy for active transport most commonly comes from ATP hydrolysis, but also sometimes from electron movement, like in mitochondria, or even light energy in some specialized cases. Scientists have classified ATP-driven active transporters into 4 major classes based on their structure and mechanism. First, B-type transporters. They autophosphorylate during their cycle, meaning they temporarily stick a phosphate from ATP onto themselves. Key examples are the Sca2 plus ATPase in muscle for calcium pumping, and the absolutely critical Na plus Kk plus ATPase in the plasma membrane. Second, F-type transporters. These are involved in energy coupling, mostly found in bacteria, chloroplasts, and mitochondria. The main example here is the mitochondrial ATP synthase, which actually runs backwards compared to most pumps. It uses a proton gradient to make ATP during oxidative phosphorylation.
So it generates ATP instead of using it.
Exactly. Third, V-type transporters, or vacuolar ATPases. These primarily pump protons into intracellular compartments like lysosomes and synaptic vesicles, acidifying them, which is crucial for their function. And finally, the ABC transporters, that stands for ATP binding cassette. This is a huge superfamily. They typically have two nucleotide binding domains for ATP and two transmembrane segments forming the pathway. They transport a vast range of things: ions, sugars, lipids, drugs. Two really important examples: the CFTR protein, the chloride channel mutated in cystic fibrosis,
We mentioned that one.
and the MDR1 protein, or P-glycoprotein. This one is notorious for pumping many anti-cancer drugs out of cells, causing multi-drug resistance in chemotherapy, a huge challenge in cancer treatment.
Among all these amazing pumps, the Na plus magic K plus magic ATP seems to be an undisputed star player involved in so many fundamental processes in our bodies. Can you dive a bit deeper into its remarkable mechanism and why it's so fundamental?
It truly is a pivotal integral membrane protein, arguably the most important active transport pump in animal cells. As we discussed, cells maintain low intracellular sodium, Na+, and high intracellular potassium, K+, plus a negative electrical potential inside. The Na+K+ ATPase is the machine solely responsible for actively maintaining these crucial gradients against their natural tendency to run down. For every single molecule of ATP it hydrolyzes, this pump goes through a complex cycle of shape changes. Specifically, it moves 3 sodium ions, Na+, out of the cell and simultaneously brings 2 potassium ions, K+, into the cell.
3 Na plus out, 2 K plus in. Why the difference?
That difference is key. Moving 3 positive charges out for every 2 positive charges can result in a net movement of positive charge out of the cell. This makes the inside of the cell electrically negative relative to the outside, which is why it's called an electrogenic pump. This directly contributes to establishing the cell's resting membrane potential, fundamental for nerve and muscle cell excitability. Structurally, it's typically made of an alpha subunit, which does the ATP hydrolysis and ion binding, and a beta subunit that helps with folding and getting it into the membrane. The ATP and Na+ binding sites are on the inside, cytoplasmic side, while the K+ binding sites are on the outside, extracellular side. The whole cycle involves binding ions, getting phosphorylated by ATP, changing shape to release ions on the other side, binding the other ions, getting dephosphorylated and changing shape back. It's intricate.
A real molecular machine.
Absolutely. And it consumes a huge chunk of cellular ATP, maybe 30–40%. Its activity is often coupled to other transport systems, like glucose uptake, which rely on the Na+ gradient it creates. And clinically, it's the target of drugs like digitalis, used for heart failure, which inhibit the pump. So, if a cell needs to move very specific molecules, these dedicated transporters act like their bespoke delivery systems, right? Each with a specific route and a particular cargo it's designed to handle. Precisely. And we can classify these transporters based on what they move and how. The simplest is a uniport system. Moves just one type of molecule bidirectionally, depending on the concentration gradient, like glucose transporters moving glucose in or out. More complex are the cotransport systems, where the transfer of one solute is coupled to the transfer of another. Often the energy from moving one solute, like Na+, down its gradient, drives the movement of another solute against its gradient,
Using one gradient to power another.
Exactly. Within cotransport, you have 2 types. Symport systems, or symporters, move 2 different solutes in the same direction. Classic examples are the Na plus sugar transporters, like SGLT1 in the intestine and kidney, and Na plus amino acid transporters. The movement of Na plus down its steep gradient, made by the Na plus MAM plus K plus ATPase, provides the energy to drag the sugar or amino acid along with it into the cell, even against its own gradient. This is called secondary active transport. Second, antiport systems, or antiporters, move 2 different molecules in opposite directions. A vital example is the Na plus mancha Ca2 plus antiporter, which moves Na plus in while pumping Ca2 plus out. This is crucial for keeping intracellular calcium levels very low. These classifications just highlight the sophisticated strategies cells use to manage their traffic.
You mentioned earlier that transporters act a bit like enzymes, sharing some key characteristics. That's a fascinating parallel. How so? What are those shared features?
It's an insightful comparison because both facilitated diffusion and active transport systems do share several fundamental characteristics with enzyme action. Understanding this helps grasp their behavior. Firstly, they exhibit specificity. Just like an enzyme binds a specific substrate, transporters have highly specific binding sites for the solute they move. They can distinguish between closely related molecules. A glucose transporter won't usually grab fructose, for instance. Secondly, transport is saturable. Like enzymes, there's a finite number of transporters in the membrane. Once they're all busy, Increasing the solute concentration won't speed things up further. This gives rise to a maximal rate of transport, or Vmax, just like an enzyme's Vmax. This saturation is a hallmark of protein-mediated processes, unlike simple diffusion.
So there's a speed limit based on how many transporters there are.
Exactly. Thirdly, there's a characteristic binding constant, coram, for the solute, analogous to an enzyme's coam. It reflects the transporter's affinity for its solute. Lower coam means higher affinity, working well even at low concentrations. And finally, transporters can be blocked by structurally similar competitive inhibitors. Just like an inhibitor can block an enzyme's active site, similar molecules can block a transporter's binding site. However, a crucial difference: transporters generally don't chemically modify their substrates, they just move them across the membrane. They're sophisticated traffic controllers, not chemists, but using similar principles.
And hormones can even get involved in regulating these transporters. That's another fascinating layer of control, bringing the entire body's signaling into play.
Absolutely. Hormones provide a powerful way for the body to rapidly adjust cellular transport capabilities in response to physiological needs. They can increase or decrease the number of available transporters on the cell surface. A prime example is insulin. In fat cells and skeletal muscle cells, insulin significantly increases glucose transport. How there's O. It triggers a signaling pathway that causes GLUT4 glucose transporters, which are normally stored inside the cell in vesicles, to move to the plasma membrane.
So insulin calls more transporters to the surface.
Precisely. By increasing the number of GLUT4 transporters at the surface, insulin raises the Vmax for glucose uptake, allowing these cells to absorb much more glucose from the blood, especially after a meal. Crucial for blood sugar control. Similarly, glucocorticoid hormones, like cortisol, enhance amino acid transport into liver cells. These amino acids are then used for gluconeogenesis, making new glucose during fasting or stress. Growth hormone increases amino acid transport in most cells, supporting growth. Even estrogens influence amino acid transport in specific tissues like the uterus. These examples really show how the endocrine system finely tunes cellular metabolism by modulating membrane transport.
Moving on to ion channels, these seem to allow for much faster passage, like express lanes dedicated purely to ions. How do they achieve such speed and still maintain their crucial selectivity?
That's a great way to put it. Ion channels are transmembrane proteins forming highly selective pores, acting as incredibly rapid conduits. They're typically very narrow, just 5 to 8 angstroms wide. Their key characteristic is high selectivity. Most permit passage of only one specific type of ion, Na plus K plus A2 plus A or Cl. Selectivity depends on the ion size, its hydration shell, and its charge density. A fantastic example is the K+ channel. It lets potassium ions flow through maybe 10,000 times faster than sodium ions, even though sodium is smaller.
How does that work?
Its selectivity filter, a critical narrow region, is lined with precisely positioned carbonyl oxygen atoms from the protein backbone. These oxygens perfectly mimic the hydration shell of a dehydrated K+ ion, allowing it to shed its water coat and interact favorably as it passes through. But a smaller Na+ ion can't interact correctly with these carbonyls in the right way. Can't shed its water easily enough or fit snugly, so it faces a high-energy barrier and is rejected. It's an incredibly precise molecular sieve, allowing astonishingly rapid K+ conduction.
So, if they're like express gates, what makes them open and close? It's not like they're just permanently open doorways. There must be a sophisticated control mechanism.
You're absolutely right. They aren't permanently open. Ion channels are highly regulated and are gated, meaning they open and close transiently in response to specific stimuli. This gating is fundamental. There are several main control mechanisms. First, ligand-gated channels. They open when a specific molecule, a ligand like a neurotransmitter, binds to a receptor site on the channel. Think of neurotransmitters at synapses opening channels to pass the signal along. Second, voltage-gated channels. These open or close in response to changes in the membrane potential, the voltage across the membrane. Crucial for nerve impulses and muscle contraction. Research shows they have voltage sensors, basically charged protein segments or paddles, that move within the membrane when the voltage changes. This movement is mechanically linked to the gate, opening or closing it. This is how action potentials propagate.
So changes in electricity open the gate.
Exactly. And thirdly, there are mechanically gated channels. These respond to physical stimuli like pressure, stretch, or touch. Channels in your skin's touch receptors open when deformed, sending a signal. This diverse control allows cells to respond dynamically to a huge range of cues.
Are there other ways ions can get across, perhaps less controlled or even problematic ways that disrupt the cell?
Yes, there are, and some can indeed be highly problematic. One category involves ionophores.
These are small, often cyclic organic molecules, frequently made by microbes that act as shuttles for ions across membranes, bypassing normal control. For instance, valinomycin specifically shuttles potassium ions, K+. It grabs K+ in its hydrophilic center and then, being hydrophobic on the outside, diffuses across the membrane. Others, like gramicidin, form hollow channels themselves. While useful in research, uncontrolled action in the cell is disruptive. On a more problematic note, certain microbial toxins can severely disrupt membrane integrity by creating uncontrolled pores. For example, diphtheria toxin or components of the activated serum complement system can produce large, non-selective pores, causing vital molecules to leak out and leading to cell damage or lysis. Bacterial toxins like iglycin from Staph aureus assemble into a barrel structure, forming large pores. These let things like ATP leak out, killing the cell. So while physiological channels are regulated, these toxins represent uncontrolled bypasses, highlighting the importance of membrane integrity.
And what about water itself? We discussed its surprisingly high permeability through the lipids, but does it have a special channel or does it just kind of squeeze through the lipids alone?
While water does move by simple diffusion across the lipid bilayer, this movement is often significantly augmented by specialized channels in many cells. These are called aquaporins. They are tetrameric transmembrane proteins. 4 identical subunits come together, each forming a water-conducting porin. Humans have at least 10 distinct aquaporins, AP1 to AP10, found in different tissues. These aquaporins are crucial because they facilitate remarkably rapid water passage while strictly excluding ions and, importantly, protons.
How do they stop protons getting through?
It's clever. Their pores are too narrow for hydrated ions. Protons are excluded by specific asparagine residues lining the channel. These bind the water molecule's oxygen in a way that prevents it from participating in the proton relay, or hopping, that normally allows protons to move through water. This ensures they are pure water channels, maintaining crucial proton gradients. Their importance is clear from genetic disorders. Mutations in aquaporin 2 cause one type of nephrogenic diabetes insipidus, where the kidneys can't concentrate urine, leading to severe dehydration. Even water movement is tightly regulated.
So that covers small molecules and ions and even water. But what about truly gigantic cargo, like entire proteins or even whole viruses? How do cells manage to take those in or release them? It can't be through a channel, surely.
You're absolutely right. It requires entirely different, highly sophisticated mechanisms. Cells have evolved remarkable processes for taking up and releasing macromolecules, things far too large for channels or typical transporters. These bulk transport processes are endocytosis and exocytosis. Both involve dynamic, dramatic reshaping of the plasma membrane through the formation and fusion of membrane-bound vesicles. Let's first look at endocytosis, how cells ingest things. A segment of the plasma membrane invaginates, fold inward, enclosing some extracellular fluid and contents. This pouch then pinches off to form a vesicle inside the cell. These vesicles often fuse with lysosomes, the recycling centers containing digestive enzymes.
So the contents get broken down.
Yeah. Exactly. Digested into smaller components the cell can reuse. This whole process requires energy, ATP, calcium, and cytoskeletal elements like actin. There are 2 general types of endocytosis. Phagocytosis, cell-eating, occurs only in specialized cells like macrophages. It involves ingesting large particles like bacteria, viruses, or cellular debris. Macrophages are incredibly active phagocytes. The other type is penocytosis, cell drinking, a property of all cells, for uptake of fluid and dissolved contents. It has 2 forms. Fluid phase penocytosis is non-selective. Uptake is just proportional to the concentration outside. It samples the environment. Cells doing this actively must constantly recycle membrane back to the surface. The second, often more significant type is absorptive penocytosis, or receptor-mediated endocytosis. This is highly selective and efficient for specific macromolecules. So it targets specific things.
Yes. It relies on high-affinity receptors on the plasma membrane that bind and concentrate specific ligands from the medium. These vesicles form from coated pits lined on the inside by proteins, most famously clathrin. Clathrin forms a basket-like cage, and proteins like dynamin, which uses GTP energy, help the vesicle pinch off. A classic example is the uptake of LDL, carrying cholesterol, via the LDL receptor.
Ah, the cholesterol uptake mechanism.
Right. But problematically, many viruses, hepatitis, polio, AIDS, COVID-19, exploit this very mechanism to enter cells. They hijack the system. Excessive iron uptake can also happen this way. Now, exocytosis is the reverse process cells releasing macromolecules. It's involved in membrane remodeling, as vesicles carrying newly made proteins and lipids from the ER and Golgi fuse with the plasma membrane, adding components to the surface. The signal for release is often a hormone triggering a transient rise in intracellular calcium. This Ka2 plus rise triggers vesicle fusion. Released molecules can become membrane proteins, extracellular matrix components like collagen, or signaling molecules like insulin or neurotransmitters. It's fundamental for secretion and communication.
So we've covered the general mechanisms. What does all this incredibly intricate transport machinery mean for something as common and vital as glucose, our primary fuel source? How does glucose navigate this complex membrane world?
Glucose transport is a beautiful integrated example, showcasing tissue specificity and different mechanisms. Glucose entry is the first rate-limiting step in its use. Different glucose transporter, GUTs, exist in various tissues. In apocytes, fat cells, and skeletal muscle, as we mentioned, insulin dramatically enhances glucose uptake by recruiting GLUT4 transporters to the cell surface, increasing the Vmax in transport. The situation in the small intestine, where we absorb dietary glucose, is even more complex and illustrates a fascinating interplay. Here, glucose and sodium ions, Na+, bind simultaneously to a Na plus glucose symporter, SGLT1, on the apical surface facing the gut.
The symporter we talked about, moving both in the same direction.
Exactly. 2 NOD plus ions move in for each glucose molecule. This is secondary active transport. The energy comes from NO plus moving down its steep electrochemical gradient, a gradient maintained by the NO plus NADH K plus ATPase pump on the opposite basolateral side of the cell. The downhill movement of NO plus effectively drags glucose into the cell, even against its own concentration gradient. Intestinal glucose uptake absolutely depends on that NO plus gradient. Once inside the intestinal cell, glucose concentration is high. It then exits across the basolateral membrane, facing the bloodstream, via facilitated diffusion, using a glucose uniporter like GLUT2.
So active transport in, facilitated diffusion out.
Precisely. GLUT2 handles high glucose loads efficiently. This intricate transcellular movement is why oral rehydration therapy, using NaCl and glucose, works so well for severe diarrhea like cholera. The coupled transport of glucose and sodium pulls water into the cells via osmosis, rehydrating the patient. It's a beautiful physiological coordination.
Beyond just moving molecules around, membranes are also highly sophisticated command centers for communication. They're the literal interface where cells receive and send critical information, effectively allowing them to talk to each other and their environment.
That's a profound observation and absolutely central to understanding multicellular life. Membranes are pivotal to this intricate information highway. Specific biochemical signals, neurotransmitters, hormones, growth factors, immunoglobulins typically don't enter the cell directly. Instead, they bind to integral transmembrane receptor proteins on the cell surface. These receptors act like molecular antennae. Their extracellular domains recognize and bind the signal. This information is then transmitted across the membrane to the cytoplasm in a process called transmembrane signaling, or signal transduction.
Relaying the message inside.
Exactly. This often involves generating second messenger signaling molecules inside the cell, small molecules like cyclic AMP, cAMP, calcium ions, Ca2+, phosphoinositides like IP3, and diacylglycerol, DAG. These amplify and relay the signal. Furthermore, many steps involve phosphorylation events, where kinases add phosphate groups to proteins, acting like molecular switches, turning proteins on or off to orchestrate the cell's response. It's a highly sophisticated system.
But some cells don't even bother with external signals and receptors. They just directly connect, almost like internal phone lines, sharing information instantly from one cytoplasm to the next.
That's exactly right, a very different strategy. These direct connections are facilitated by specialized structures called gap junctions. Gap junctions allow the direct transfer of small molecules up to about 1,200 Daltons, including ions, small metabolites like ATP, signaling molecules like KMP, directly from one cell cytoplasm to its immediate neighbor.
Wow, direct sharing.
Yes, allowing very rapid communication and coordinated activity among groups of cells. Critical in tissues like the heart or epithelia. Structurally, gap junctions are made of proteins called connexins. 6 connexins assemble to form a hemichannel, or connexin, in one cell membrane. This hemichannel then aligns and joins with a similar hemichannel in the adjacent cell to create a complete, membrane-spanning connexin channel, bridging the two cytoplasms. Many connexins cluster together to form a gap junction plaque. Different connexins are found in different tissues, and mutations in their genes are linked to various conditions, including cardiovascular issues, certain types of deafness, and a neurological disorder called Charcot-Marie-Tooth disease. Direct sharing is crucial.
Now, this is where it gets really interesting and is at the forefront of biological discovery. There's a relatively new area that's fundamentally changing how we think about cell communication, almost like cells are sending out tiny information-packed letters or biological USB drives.
You've perfectly captured the excitement. In just the last decade or so, a class of secreted vesicles, broadly termed extracellular vesicles, EVs, has been recognized as a significant, previously underappreciated mediator of cell communication. They are implicated in both normal physiology and many diseases. What's key is their structure, enclosed by a lipid bilayer carrying a distinct payload of molecular information from a source cell to a target cell. True intercellular messengers.
That's incredible. So how exactly do these tiny biological messengers form? Are they all made the same way?
That's a critical distinction. There are at least two distinct mechanisms. One class is microvesicles. These form by direct outward budding and pinching off from the plasma membrane of the source cell. They're typically larger, maybe 100 to 1,000 nanometers. Their content reflects the plasma membrane and cytoplasm nearby. The other major class, perhaps most studied recently, are exosomes. They have a more complex origin, forming from multivesicular bodies, MVBs, which are internal endosomal compartments. Inside the MVB, smaller vesicles, the future exosomes, bud inward into its lumen. When the MVB fuses with the plasma membrane, these internal vesicles are released outside as exosomes.
So they form inside first, then get released.
Exactly. Exosomes are generally smaller, maybe 30 to 150 nanometers. In both cases, these released vesicles then interact with or are taken up by a target cell to deliver their payload.
And what kind of payloads are we talking about here? This really does sound like a biological USB drive.
It's a great analogy. The content is incredibly diverse and varies depending on the source cell and its condition. They carry proteins, cytoplasmic proteins, nuclear proteins, and many membrane-bound proteins like receptors, channels, MHC molecules. But maybe even more fascinating is the nucleic acid cargo. They've been shown to contain DNA, genomic fragments, mitochondrial DNA, messenger RNA, mRNA, which can be translated in the recipient cell.
Wow, instructions.
Exactly. And a plethora of non-coding RNAs, ncRNAs, like microRNAs, miRNAs, long non-coding RNAs, circular RNAs. These ncRNAs, especially miRNAs, can profoundly regulate gene expression in recipient cells. They also carry lipids and metabolites. Given this richness, it's not surprising they're implicated in a vast range of processes and diseases: immune responses, tissue repair, nervous system communication, cancer progression, metastasis, drug resistance, neurodegeneration. Because they can potentially target specific cells due to their surface proteins and carry diverse cargo, they're attracting huge interest for therapeutic delivery drugs, genes, maybe even CRISPR components. It's a very dynamic field with immense potential.
Given how intricate, dynamic, and absolutely vital membranes are, controlling everything from internal balance to external communication, it's perhaps not surprising that defects, even subtle ones, can lead to a whole host of profound problems and severe diseases.
Indeed, it's fundamental. Mutations affecting membrane proteins or disruptions to membrane lipids can cause numerous diseases. While some mutations directly impair the protein's function, like breaking an enzyme or a channel, the majority of disease-causing mutations actually cause protein misfolding. This misfolding then disrupts their proper trafficking from where they're made in the ER to their correct functional location, like the plasma membrane or lysosomes.
So they get made, but they don't get to the right place, or they're folded wrong when they get there.
Exactly. It's not just about what they do, but if they even get to where they're supposed to do it, and if they're functional when they arrive. This trafficking defect concept is central to many membrane-related diseases.
Let's look at some specific examples from our sources, really digging into the details, to truly bring home the diverse and devastating impact of membrane abnormalities.
We can draw from many well-documented cases, for instance, looking at receptors. Achondroplasia, the common form of dwarfism, involves mutations in the fibroblast growth factor receptor 3, FGFR3, which regulates bone growth. The mutation makes the receptor overactive, stopping bone growth too early. Familial hypercholesterolemia, FH, results from mutations in the LDL receptor gene, impairing the cell's ability to take up LDL cholesterol from the blood via receptor-mediated endocytosis, leading to high cholesterol and heart disease. In transporters, Wilson disease involves toxic copper accumulation due to mutations in ATP7B, a copper transporter needed to excrete copper. Cystic fibrosis, CF as we'll discuss, is caused by mutations in the CFTR protein, a chloride channel transporter. For ion channels, congenital long QT syndrome, a heart rhythm disorder, is often caused by mutations in heart ion channels, potassium or sodium, affecting the heart's electrical cycle. With membrane-associated enzymes, ISIL disease is a lysosomal storage disorder caused by mutations in GLC and phosphotransferase.
This enzyme normally tags lysosomal enzymes for delivery to lysosomes. Without the tag, the enzymes get secreted outside, and waste builds up inside lysosomes. Defects in structural proteins. Hereditary spherocytosis involves mutations in red cell membrane proteins, like specrin or ankyrin, weakening the membrane skeleton, making red cells fragile and spherical. Issues with glycosylation, adding sugars. Abnormalities in sugar chains on membrane proteins and lipids are thought to play a role in cancer metastasis, affecting how cancer cells interact and spread, and problems with protein anchoring. Paroxysmal nocturnal hemoglobinuria, PNH, results from a deficiency in attaching GPI anchors to certain red cell proteins. These proteins normally protect red cells from immune attack, complement. Without them, red cells are destroyed. These examples clearly show defects across all membrane component types can cause specific diseases.
Beyond these genetic mutations, are there other ways membranes can be affected? Can external factors, toxins, or other cellular processes directly disrupt their function?
Yes, absolutely. While genetics are key, membranes can also be compromised by other factors, autoimmune diseases, for instance. In myasthenia gravis, the body makes antibodies against the acetylcholine receptor at the neuromuscular junction, impairing nerve-muscle communication and causing weakness. Environmental stresses matter too. Ischemia, lack of blood supply, rapidly compromises ion channels and pumps. In a heart attack, lack of oxygen impairs the Na plus K plus AT pace, leading to ion imbalance, swelling, and cell death. In cancer therapy, overexpression of the P-glycoprotein, MDR1 pump, is a major problem. It pumps chemo drugs out of cancer cells, causing multi-drug resistance, MDR, and making treatments ineffective. And abnormalities in membrane lipids themselves can cause issues. Excess cholesterol, like in FH, alters fluidity and protein function. Lysophospholipids, formed when a fatty acid is removed, maybe by snake venom or cell stress, can act like detergents, disrupting membrane integrity. And accumulation of specific glycosphingolipids and sphingolipidoses like Tay-Sachs disrupts membrane organization, causing severe neurological problems. So both proteins and lipids are critical and interdependent.
Let's zoom in on one particularly significant example that profoundly touches on many of these points. Cystic fibrosis. How does a defect in a single membrane protein lead to such a systemic and severe disease?
Cystic fibrosis, CF, is indeed a profound and devastating example. It's a serious recessive genetic disorder, relatively common among people of white descent. Its hallmarks affect several organ systems, mainly epithelial tissues. Patients typically have chronic bacterial infections in airways and sinuses, leading to progressive lung damage, the main cause of death. They often have fat maldigestion due to pancreatic insufficiency as ducts get blocked by thick mucus. Male infertility is common, and a key diagnostic sign is very high chloride levels in sweat.
Sweat test, right?
Exactly. The underlying cause is mutations in a single gene on chromosome 7, encoding the cystic fibrosis transmembrane regulator protein, or CFTR. CFTR is a unique ABC transporter, but functions mainly as a cyclic AMP-regulated chloride channel. It moves chloride ions across the apical membranes of epithelial cells in airways, sweat glands, pancreas, etc. When CFTR is dysfunctional due to mutation, the most common one, FAIF508, causes misfolding and prevents it from reaching the membrane. Chloride transport is severely impaired. This impaired chloride transport causes a cascade of problems. Since chloride can't exit properly, water doesn't follow by osmosis. This leads to thick, sticky, dehydrated mucus in lungs, pancreas, etc.
And that thick mucus causes the problems.
Precisely. It clogs airways, creating an environment for chronic infections, especially Pseudomonas, and blocks ducts in the pancreas and elsewhere, causing digestive issues and other symptoms. It's a tragic illustration of how a defect in folding, trafficking, and function of one crucial membrane protein has systemic, life-threatening consequences. Therapies targeting the CFTR protein itself, called modulators, have been a major breakthrough. #outright.
Wow, what an incredible journey. We've literally gone from the tiniest lipid components that spontaneously self-assemble into that fundamental barrier, all the way to the incredibly complex protein-studded machinery that powers transport, communication, and, well, overall cellular integrity. We've seen how membranes are not just passive boundaries, but dynamic, active, and utterly indispensable participants in every single aspect of cellular life.
Our deep dive has indeed revealed membranes as far more than just simple cell borders. They are dynamic, asymmetric, protein-studded powerhouses, meticulously orchestrating everything from establishing crucial ion gradients and facilitating nutrient uptake to mediating complex signaling pathways and enabling direct intercellular communication via gap junctions. The depth and complexity are truly astounding.
Understanding the elegant simplicity of the fluid mosaic model, the nuanced differences between passive and active transport, and that incredible intricate dance of lipids and proteins. really gives you a foundational, profound mastery of cellular life. You can now deeply appreciate just how critically normal, perfectly functioning membranes underpin normal cellular function, and how even subtle changes can cascade into widespread disease. You really do now possess the knowledge to decode this essential cellular architecture.
And as you continue to observe the world, perhaps consider the ongoing, rapidly accelerating research into extracellular vesicles, particularly exosomes. Given their remarkable capacity to carry such diverse payloads, DNA, mRNA, non-coding RNAs, and their demonstrated ability to specifically target recipient cells. How might these tiny membrane-bound packages revolutionize future diagnostic approaches, maybe acting as biomarkers, or entirely new therapeutic strategies for diseases we've traditionally struggled to treat effectively? It's a question that really challenges current paradigms and offers immense promise.
Something profound to think about as you go about your day, knowing the intricate, dynamic, and absolutely essential world thriving within and around every single one of your cells. Until next time, keep diving deep.